Oh, yeah, so yeah, you did the how. Okay, so I think you knew this. And then like I'll do the argument. Yep. If you want to chime in. Okay, sounds good. Yes. Yeah, we're ready. All right, I guess we're all ready to go. Thanks for your patience as we kind of transition in between computers. So thanks for coming. It's a good crowd today, so hopefully you're gonna get a lot out of this seminar. My name is Dr. David Taylor. I'm in the marine biology department.
I'm Dr. Katie Matini. I'm also in the biology department.
So this is a seminar we've given for a number of years now, and it's really the intent is to kind of ask questions about why is it important to get involved in research, how do you go about getting involved in research, and then we're going to explore numerous opportunities that exist on campus and off campus for those research opportunities. There's a few things that I wanted to mention at the start, just kind of things to think about as we go through these presentations. Just out of curiosity, how many of you are first-year students? Wow, great, a decent number. Okay, so one of the things that I like to communicate to the students is when you're looking for research opportunities, start early. So all because you're a first-year student, I don't want you to think that you have to wait or buy time until you actually get that research experience. It's oftentimes, myself included, many other faculty, when we have long-term projects, meaning we know that they go on for multiple years, when we recruit students into our labs, we intentionally seek younger students, maybe first-year students, maybe second-year students, knowing or recognizing that you're probably going to work with us for a number of years. If it takes maybe a little bit of a learning curve of what we're doing in our lab, maybe a semester, maybe a year. If you were to join us, say your junior year or senior year, by the time that you know what's going on, you've moved on, and really you haven't benefited much nor as the faculty. So if you're early in your career, you kind of get that learning curve out of the way, then you can be really productive for 2 or 3 years. So start early. Okay, start early. The second thing I wanted to mention are many of the opportunities that we're going to present have very specific deadlines, okay, for applications or proposals. Sometimes that's within a given semester, sometimes that's within a given year, and sometimes they're very specific to year class. For example, you might be eligible as a sophomore, but no other time. So be aware of that as well. And last but not least, the third thing I want to mention is there's going to be a lot of information coming at you in this presentation. And you might feel the need to kind of feverishly write everything down, and you might be upset if you've missed something. Fear not, this is being recorded, right? And the link will be provided to you. And the other thing I'll mention is that if you're interested in the PowerPoint slides, if you don't see them that well in the video, certainly you can reach out to me and I can send you a PDF copy of the PowerPoint slides. And there's also some documents where there is a document that outlines or identifies the faculty in the Division of Math and Science, and it identifies their research interests and potential student projects. Have you all received that in an email?
Okay. Taylor, we can also post it on the M&S seminar website.
Okay, great. So does that sound good? All right, so this right here is the schedule that we're going to be working through. We're going to have a presentation by myself and Dr. Matini talking about, again, how to get involved in research. Then you're going to see prime examples of student research, both in the form of oral presentations. We're going to have 3 student speakers, and then we're going to have a poster session out in the lobby that follows after that. So that's how we're going to kind of run the show. So with respect to the presentation that we're delivering now, how we're going to break out opportunities are two ways. One, there are on-campus opportunities, and the second are going to be the off-campus opportunities. And then I'll be talking about the on-campus opportunities, and there I'm going to split them out for opportunities that exist during the academic year, and then opportunities that exist over the summer. Okay? And then you can see some of the off-campus things we're going to talk about as well. So we're first going to start with this. Why participate in undergraduate research? And I'm not going to give you the answer. I'm going to ask you, why are you here? Why are you in this room right now? Why do you think it's important, or why do you want to get involved in research? What's your name?
Marina.
Maria?
Marina.
Oh, Marina. Okay, Marina.
To better understand what I'm researching or what I'm studying.
Okay, love it. All right, I love it. The way I like to think about it is you're going to have a lot of coursework involved with, obviously, with being a student here, applying what you've learned in coursework to kind of hands-on experiences. So I really agree with that. Anybody else? Yeah, what's your name?
Matthew.
Matthew?
Or Matt is fine.
Matt, okay.
But I mean, I've wanted to go into like, especially marine research for like basically my whole life.
Okay,
I did an internship over the summer at Columbia in New York City
okay.
at Columbia University, and that only further like solidified that as what I want to do.
Okay, so you're fulfilling a life promise.
Yes.
That's fantastic. Okay, anybody else? Yeah, what's your name in the back?
Ariana.
Ariana?
I want to improve my lab techniques,
Okay.
and I want to gain the skills so I can start my own research project in the future on
Okay.
viruses,
Right.
viruses.
Fine tune your skills, maybe get some insight into developing your own projects. Awesome. Anybody else? No, what's your name?
Sarah.
Sarah?
Make up for lab time lost during
Oh, that's a new one. That's a great idea. So to make up for lost time due to COVID. Yep. So, yes, Sam. Sam.
Yeah.
Holy cow, I got that right. I'm sorry.
Personal interest.
Personal interest. All right. Passion. You guys come on with great ones. Anybody else? Yep. What's your name?
Connor.
Connor. That's right. Connor. Yep.
To figure out what I actually want to research further on in my career.
Okay. Awesome. Understand, fine tuning what your interests are so you know what you might want to pursue later in life. Man, you guys really came up with a lot of them. So I'll show you what I have on my list, but I think you guys pretty much covered. Well, clearly research is an integral part of science. And to Marina, did I get that right? To Marina's point, you're going to have a lot of coursework, but this is your opportunity to go beyond that, even go beyond your lab courses, but apply what you've learned in these classes to unanswered questions in the form of research topics. To Connor, I believe this was your point almost, learn now if you like research, right? And you might think, yeah, I think I'm interested in research, but I don't know for sure. I'll tell you, now is the time to learn that. You don't want to go through 4 years of college, think you want to do a research project, go on to, say, graduate school to do research, never having a lick of experience. Maybe you get into a graduate school without research experience, that's tough, but let's say you do. And then you're in your first year of a master's or a PhD program and you learn that you don't want to do research. That's not a good time to learn that. You want to learn those things now. Let's also say you do understand that you want to do research. What do you want to do research in? There's a lot of different things you can do. There's research that is field-based. There's research that is laboratory-based. Maybe you want to be a lab rat, or maybe you want to work on computers and do quantitative modeling. There's a lot of flavors to research. Now's the opportunity to learn, do you want to do research? And what do you want to pursue in your research endeavors? This is one that didn't really come up, but it's important. It is a great opportunity, or it's a great experience to get you to the next step. What do you want to do after Roger Williams? I presume you want to work. I presume maybe you want to go on to graduate school, something along those lines. Research experience now is going to help you get to that level. And not only will it help you, in many respects, it's becoming a requirement that you won't get a job or you won't get into graduate school without some form of research experience. Another one here, you'll come to learn from the students that are presenting today. Many of them have presented at scientific meetings, at conferences. This is a great opportunity for you to network. I always tell my students when they're in this position, introduce yourself as an undergraduate student. Saying, is it Matt? I am Matt from Roger Williams University. I am in my X year. I'm going to be presenting on such and such topic. If there's someone in the audience that is a potential employer or a potential graduate advisor, they could be very impressed by what Matt had to talk about, and that's kind of starting to network Matt into a position. So it's a great networking opportunity. And this last one is one of my favorites to talk about. I call this, I said it last year and I say it every year. You get to experience the full spectrum of the human emotion. How awesome is that? What do I mean? Research can be fun. It can be depressing. It is exciting. Sometimes it's boring. It's often frustrating, not often, it's always frustrating. But with that comes extreme challenges and extreme reward. You can experience that in a semester. You can experience that in a day doing research. So that's what I mean by you experience the full spectrum of the human emotion. So what is better than that? Nothing. Nothing's better than that. So we all want to be researchers, right? Okay, so that was the why. Dr. Maritani is going to briefly talk about how, in this case, how to approach faculty with respect to getting involved in research.
Yeah, so one of the things, so I teach a lot of intro bio, right? I have a lot of first year students, second year students. And one of the things that I've come to learn that I don't think I appreciated when I first started teaching is that sometimes students are intimidated by coming to office hours because I'm super scary like that. Right. And so sometimes also people like students don't know how to approach like a faculty mentor. And I would say kind of in broad terms, everyone here who does research, like that's the thing they're super excited about. And if you want to talk to them about their research, mostly they're going to be super excited that you're interested, right? So I would not see it as, something where you have to be worried about it. It's really going to be something that I think most professors will be excited to talk to you about their research. Okay. So things to do if you're interested in research, first is learn about what different faculty are doing, right? Professor Taylor alluded to, we have a document that we will put on the website, and that I think has also been emailed out to you, that lists all the faculty in M&S and kind of what their research interests are, right? For all the different departments. So whether you're bio, chemistry, math, whatever, their kind of research interests are on there. So you want to peruse that and see, are there any interests that overlap with what you're interested in? There's also the Roger Williams website, right? That's probably not updated quite as frequently as this document, but there's some information just out there, if you Google it. And then if you have that faculty member in class, you can just approach them after class. Otherwise, you can send them an email. It's probably not great to just be like, hey, can I meet with you to talk about your research? You might want to put a little bit more into it. These are just some thoughts, things you might say. What about their work is interesting to you? If you did look, on that document that lists all their interests, be like, I saw that you do this thing with marine viruses, and I'm really into that. So can we talk about that some more? If you have a specific career interest, like if you already know, I really want to become a veterinarian, or I really want to go do a PhD program. Those are things that can be useful to mention. Sorry, I keep taking my glasses on and off because I have this mask and glasses problem and I can't read anything. If you have one, if you already have one, it can be helpful to include like a resume or CV, or if you're first year, you can just be like, I'm in Bio 103, or I'm in Oceanography, and so is my professor. Especially if you think your professor might say something complimentary about you. Because if I got an email from someone interested in research, I would go to Dave and be like, Dave, you have this person in class, tell me about them. That can be a helpful thing. And you can ask if there's space in their lab. Sometimes labs are full, so even if there's a good fit in terms of interests, there may not always be an immediate opening, but it could be something in the future. So ask if there's space in their lab. If they have space, you can say, hey, can we talk about this some more, right? And as I said, professors love to talk about their work. So this is an email I think most people would be pleased to receive.
And just I'll throw out tidbits of recommendations here and there. But if or when you approach faculty in an email about a research opportunity, I highly recommend that you send it to one individual at a time, and you do a little bit of homework on what that individual does. There are times where I receive an email from a student who expressed interest in research, fantastic, but they basically said, I'm interested in research, and I see that there's 8 faculty listed there. So again, I appreciate the enthusiasm and willingness to want to get engaged in research, but it was clear that they didn't take time to really understand what I do. And therefore, it might not be, I don't know if it's a good match or not. So personalize the email. Even if you do reach out to multiple faculty, that's fine. Just send separate emails and tailor it to their specific interests and hopefully your interests as well. Okay. So now moving on, so we did kind of the why and the how. Now we're going to move into kind of the what. what kind of opportunities exist. As I mentioned before, we're going to break this into two groups: on-campus opportunities, which I will talk about, both in terms of during the academic year and then also the summer. And then Dr. Matini is going to be talking about off-campus opportunities. And then what you see over here on the left-hand side in this banner here, this highlights the opportunities that we'll be discussing. And when it's highlighted in white specifically, that's something that we're talking about on that slide. So here are various academic year opportunities. And each one of these I'm going to spend a little bit of time talking about, but I'm just listing them here in order. So undergraduate research. So this is where you're actually doing research for credit. Senior thesis. This is obviously done in your senior year, but it also is credit bearing. We also have things referred to as SEED grants. SEED stands for the Center for Economic and Environmental Development. We also have a Provost Fund for student research. This provides funds for you to present at scientific meetings. And then we also have something referred to as the FIT program, or Finance Individualized Training, which is, as I understand it, a subset of a work study program, which we'll talk about. So each one of these, I'm going to spend some time talking about, but I did also want to mention the three others. One is you can do research as work study. Now, if you're not familiar with work study, it's a financial aid package where the university will grant you a certain number of hours that you can work in a semester or a year. And you can use those work study hours to work in the stock room, to be a tutor, to work at the gym, to work, in any number of places. But you can actually use work study to do research as well. So if I have a project in mind and maybe I don't have an active grant to support it, if you're a work study student, you can actually receive payment or salary to do research with me. So that's work study. Faculty external grants. Now what I'm referring to here, our faculty in our division are held to a pretty high standard with respect to scholarship, right? So we're required and we want to do research. And research costs money, so we have to apply for external grants to get money to do that research. Because there's no postdocs here, because there's no graduate students here, we bring undergraduates into our lab to help drive the research program. And because of that, almost always within the grant and in our budget, we have a line item for basically student salaries. So you can be paid off of faculty grants. And then the last one that I have here, it's maybe the most rudimentary, but volunteering or shadowing. So Dr. Matini had mentioned that perhaps you reach out to a faculty member and their lab is full. Maybe they have 4, 5, 6 students, and they're just not really taking on new students, not for credit or not for payment or something along those lines. But perhaps you're willing to get engaged in research in a volunteer or shadowing capacity. By shadowing, I mean you watch what the other students are doing in the lab. That is a great way for you to get your foot into the door. So when an opportunity does open up in that lab, you are a natural fit to move right into it, if that makes sense. So I just wanted to mention those three. While moving forward, we're going to talk about the other ones. What I'm showing you here is all of those opportunities provide you the research experience, right? And that's really what you're looking for. But also have in the back of your mind that there are some other benefits that come with research in certain instances. Some of them are credit bearing, right? So they go towards your degree requirements. Some of them you receive a salary, it's nice, and some of them provide funding, give you money to basically help support the research, maybe buying supplies, something like that. So these are the ones I'm going to talk about each individually, and then I just have a check mark if they provide you that ancillary benefit, that extra benefit, and then an X if really you're not going to get that. So have in the back of your mind, not just the research experience, but possible credit, salary, and or funding. Okay. So as promised, the first one that we're going to talk about is undergraduate research for credit. It's an actual course and it has a 450 number designation. So it could be bio 450, math 450, environmental science 450. So you're actually doing research for academic credit. You will have a faculty mentor, so you would enroll with an individual. That person would be your faculty mentor. The number of credits that you can take ranges between 1 and 3 credits. And generally speaking, 1, 2, or 3 credits amounts to 3, 6, or 9 hours of research per week. Right? So obviously, the more credits, the more research you're committed in that semester. There's also a difference in the products that are produced at the end. 1 credit, there's lesser products that are produced. 3 credits, there's more products that are produced. For example, at least in my lab, if you are enrolling in 3 credits, the expectation is you will give a presentation, a public presentation of your research. Okay, so that's one example of doing research for credit. Again, the 450 designation. Another way that you can actually do research for credit is our senior thesis program. So this is obviously specific to your senior year. And in this case, not only do you receive academic credit, the designation is 451, again, Bio 451, Math 451. It's a 3-credit course, okay? And it also provides funding. You can receive up to $500 to support your research program. Again, research isn't free, so anything that you can get to support it is helpful. Once again, you are going to work with an individual faculty member. In this case, you are actually responsible for developing an independent research project. You write a proposal, okay? Have in the back of your mind, even though this is called senior thesis, the proposal is due late March of your junior year. Late March of your junior year. Okay, so late March of your junior year, you write an independent project proposal. It gets evaluated by faculty within the division. It's evaluated on its scientific rigor. It's evaluated on its fiscal responsibility if you are putting together a budget. And then you receive feedback. And then if you're given a thumbs up, you proceed with the research. Okay? I will note that many, I would say most of our students that pursue senior thesis actually are engaged in research for 2 or 3 years. So oftentimes, they're kind of pros at research at this point, and they're putting this proposal together, proposing, they're bringing in data with it, and then proposing what they're going to do in the future to put it together as a senior thesis. So here you can kind of see the various processes that are involved. Again, identify a project, identify a faculty mentor, write the proposal, get it reviewed, carry out the research. One thing that you see here, a prerequisite for senior thesis is that course, Undergraduate Research, Bio 450 or Math 450. If, however, you have substantial research going into this experience, that gets waived. You can ask for that to be waived. And then finally, in terms of products, if you're a senior thesis student, you are required to write a final written thesis, which is due towards the end of your senior year, and give a public presentation, which is almost always on a Friday in this room right here at the end. Best day ever. Best day ever. Okay, so that was senior thesis, credit and funding based. Now here I'm going to provide you some opportunities or identify some opportunities that are not credit bearing, but they are funded. And this is one that's quite nice. This is the SEED grant. So again, SEED stands for the Center of Economic and Environmental Development. Twice a year, you can apply for SEED grants of up to $1,000. And this is for research supplies. Okay? So again, what you do here is you identify a faculty sponsor. You write an independent project proposal. You again have it reviewed by the division. You get feedback. And then funding is based on the quality of that proposal. Again, $1,000 is what you can elect per proposal. I'll also note here, senior thesis, or I'm sorry, not senior thesis, SEED proposals can have joint students on it. So that doesn't have to be a single individual, even though that's the majority of the time how it works. You can have multiple students on one SEED project. And here you can see the deadlines, again, keeping track of some of the specific schedules that you have to follow. There are 2 deadlines each year, typically one each semester, early November and late March. I also just want to do an aside right here. This gentleman right here, this is Mark Gould. He is the founder of the SEED program. He's a late professor. I always like to give him his due because he's actually the founder of SEED. Another funding opportunity that I've referenced before, and I'll talk about in a little more detail. This is the Provost Fund for Student Research. And what this does is it gives you funds to present your research at a scientific meeting. Again, it can be local, it can be regional, it can be national, in some cases international. Now, in terms of the awards that they offer, I believe it's set now that you can receive up to $500 per student or $750 per project, because at times there's a project that can have more than one student on it. So $500 per student or $750 per project. And what this is going to do, again, it's for the conferences. So when you present at these meetings, there's always a registration fee that has to be covered. If you're traveling some distance, you can cover the cost of fuel, car mileage, airfare, and then hotel, things like that. So this is used to help defray the costs of what it is to go to a scientific meeting. Just like the SEED grants, this is offered twice a year. 2 deadlines, typically mid-October in the fall semester and early February in the spring semester. And then I believe the last one before getting into the summer, this is the Finance Individualized Training Program. you want to speak on this one?
Yeah, sure.
So Dr. Martini has more experience in this than I do, so I'm going to turn it over to her.
Yeah, so the FIT program is a 3-year program. It has to be the full 3 years. And so as such, you would be seeing listings for possible FIT positions kind of towards the end of your first year, right? And I think a lot of people apply for these FIT positions either at the end of their first year or kind of like over the summer as they're going to be a rising sophomore. So the faculty would write, or you could write in conjunction with them if you're working on it together, a description of kind of the plan for these 3 years. And it has to be, it's paid through work study, right? So this is only for students who are eligible for work study. And the idea is that you kind of have increasing amounts of independence and responsibility over the 3 years, right? So this is a really great situation because, as Dr. Taylor said, right, it's a lot of faculty want to get someone kind of in their early time here and then have them be able to work on in that same lab or project for many years. Other things to say, there are some like workshops that are associated with it, as well as the research aspect, and there's salary. So all of those are great things.
All right, thank you. All right, so moving on to summer opportunities. And this is really when a lot of us get the vast majority of our research accomplished. Again, you guys are busy with classes, we're busy teaching classes, committee work, but it seems like the summer is when we really get a lot of our work done. And lo and behold, there's some opportunities that exist for you. So this is what I'm just saying is summer research at RWU and other Rhode Island institutions. So again, some of these programs, you can be a Roger Williams student, but work at another institution, whether it's Brown, URI, Providence College. So there's 3 that I'm highlighting here. The Mark Gould Memorial Research Fellowship. So you remember the gentleman that I had pictured down to the bottom right? We have a fellowship in his honor. In this case, if you submit a seed proposal and or if you submit a senior thesis proposal, and that work is going to be conducted over the summer, you can ask to be considered for the Mark Gould Fellowship. We typically award 1 per summer. We also have summer, what we refer to as our SURF program, our Summer Undergraduate Research Fellowships. And these are principally supported by NIH and NSF. And really what it is, it's called, one of them is referred to as an IMBRI program, and one of them is referred to as an EPSCoR program. Not to get into too much detail here, but what this represents are states like Rhode Island that historically have not received a lot of federal funds and grants. They are open to having this program to them. So all institutions in Rhode Island are part of this. NIH IMBRI tends to be more biomedical in nature, and NSF EPSCoR tends to be more ecological in nature, particularly with marine biology and marine ecology. So you could, as a Roger Williams student, apply for a SURF fellowship and work at Roger Williams. Or there could be a project that is presented at another institution that you apply for and get and work at Brown or URI for the summer. And then finally, again, I mentioned that most of our research gets done during the summer, and how we achieve that oftentimes is through having our own, that is faculty, external grants. As I mentioned before, you can be supported by those. What comes with these opportunities? Generally speaking, regardless of which opportunity I'm talking about, it comes with a nice stipend. Right now, it typically ranges between $5,000 and $5,500 per summer. So not too bad. So $5,000, $5,500 per summer. Also, if you are a full-time summer research student at Roger Williams, and by that I'm defining it as 10 weeks, 35 hours of research per week, you are offered housing. You are offered housing, and that would be over at Bayside, if you're familiar with that. So Bayside, so you get that as well. In terms of funding, is funding available to you? That would depend on the program. That would depend on the program, but typically, in many instances, yes, there is funding available to you. In this case, I already defined what it would mean, 35 hours of research per week for 10 weeks. And then oftentimes, particularly if you're a SURF fellow, but many of our summer students do this, you will be presenting your research at a conference at URI, an undergraduate conference at URI at the end of the summer, or typically late July. Usually the program would go late May to late July. That's the 10-week time frame. Okay, here I'm just showing you, not to, you have to scribble down. Oh, I'm sorry.
You can tell my sort of life, but that's what you're telling me.
I'm sorry?
When to apply for those, is that the next slide?
This, yeah, it's outdated, hasn't been updated, but I was going to show them the website that outlines a lot of these. So again, don't worry about writing down this website. I believe it's active. I believe that's where I actually grabbed this. You can see this is actually from last year, but if you go to this website, and again, you can get it through the video or you can ask for the PowerPoint slides. If you go to this website, this outlines a lot of the research opportunities, and it goes through some of the experiences that I just mentioned and how you go about applying. Is there any extra detail that you would want to add to
Well,
this?
see, the solar server, excellent, we have lots of students who do that, but the deadline, it comes up really quick in February. And it's an external deadline, right? I mean, you have to apply on a website that's sponsored by URI for both the INBRI and for the F-score. And so the problem really is that people think about it, they send it out during January and you're all or somewhere else in January. So if you're interested in working in the CERT program, watch for that in January so you can apply like the first weekend in February.
Yeah, so how it actually works is the faculty at all the institutions will first receive an email saying, are you interested in hosting a CERT fellow? And if you are, please write up a short description of what the project would be. So then they accumulate all of those projects from all the institutions and they organize them. And once they're all organized, they then open it up to you saying, are you interested in a CERT fellow? And that, to Dr. Marston's point, will be opened up around like mid-January. In that case, you have to have letters of recommendation. You have to fill out a very brief application form. And then what you do, I think transcripts are involved, things like that. And then you select, I believe, your top 3 projects, whether it's Roger Williams or another institution. And if you're selected as a fellow, they do their best to match it up. And you might wonder, what is it like here in the summer? It's awesome. That's what I'm going to say. It's a very vibrant community in the summertime, meaning there are a lot of students here, many taking classes, but just this building itself is very active with research. It's not uncommon for us to have 40, maybe upwards of 50 students, maybe closer to 40 students in the summer engaged in full-time research. So particularly end of May, end of July, it's a busy place. It's just busy in a different way. Not so much classes, but just research. Whether it's field-based, whether it's lab-based, whether it's computer modeling, it's a really dynamic time. August is a little more chill. Most, I'm still here, but a lot of students are gone at that point. Many faculty are off for some time.
But to Professor Marston's point, I think, when I was an undergrad, applying for summer research fellowships was always my project over like winter break, right? And a lot of people aren't necessarily thinking about summer yet over winter break, but if you want to do research, that's when you should be thinking about it.
Absolutely. Okay. I think that concludes what I was going to talk about, and we're going to transition over to off-campus opportunities.
Great. So I'm going to talk a little bit about applying for various opportunities outside of Roger Williams, where you can do research in other places. One thing to note with these is that a lot of them are quite competitive, right? And so many times you need some type of research experience beforehand to be awarded one of these opportunities. So for instance, I was an undergrad at Providence College many years ago, and I did research at Providence College for a couple of years and then applied somewhere else and went like to the Mayo Clinic in Minnesota and did research there for a summer, right? But I probably wouldn't have gotten that position unless I had some experience already. So if you're interested in one of these outside positions, it probably still behooves you to start thinking about doing something here ahead of time. Basically, like we've been saying, I think over and over, start looking early. This could be a winter break project to start, looking at what the opportunities are and putting an application together. And I'm going to talk about several different programs here. And as usual, they all have very slight variations in what they want and when their deadlines are. So just read the instructions carefully. Obviously more about the NOAA program.
Sure.
So do you want to jump into
Yep.
this one?
So this is something that we've had a number of students participate successfully in. This is the Ernest Hollings program, or just Hollings is what it's referred to as. It's actually a 2-year scholarship, full scholarship. And it's offered your junior and senior year. However, the application is due explicitly in your sophomore year, at the end of January. So you got one crack at it. Okay, one crack at it to apply. And it's a very lucrative fellowship. You can see what it provides in terms of academic assistance, nearly $10,000 per year. It is a 10-week internship at a NOAA facility during the summer. And this would be the summer of your rising senior year, so in between your junior and senior year. So you receive financial aid for your junior and senior year, and you have this incredible research experience sandwiched in between those years. It also provides travel funds and housing subsidy. And again, you might think, Noah, that this is going to be exclusively oceanic or atmospheric sciences. It's actually more broad than that. It's more broad than that. And we may have had, I'm hazarding a guess, in the last, well, since I've been here, maybe 10 students. Does anybody know? A decent number of students that have actually been awarded this, maybe even more. This is not nearly the most recent, but this is Sadie Crosby, who I like. She's actually doing electrofishing in freshwater environments, and I like doing that, so that's why I have that picture.
Okay, so Professor Taylor talked about some of the opportunities for summer research, both at Roger Williams and at other institutions in Rhode Island. Believe it or not, there are places you can do research outside of Rhode Island as well. And so there are a couple of different types of programs where you can look at to try to find these. So one of them is through NSF, the National Science Foundation. And these are like kind of generally referred to as RUs, Research Experience for Undergraduates. And I don't think we have the link on here, but if you just Google NSF RU, you'll come to their webpage. And they have this huge kind of database of different opportunities that are available during the summer, right? You can filter by, oh, I need to stay in my home state, so I want to look geographically. You can filter obviously by what type of field you're in, right? So if you want to do chemistry research, you can filter by that. Yeah, and it's across all the STEM disciplines, right? So this is broad. Again, applications kind of in winter, early, kind of beginning of spring. Each university sets their own guidelines, right? So this is something, the programs are funded by NSF, but the different universities host the programs, right? So you might be going to like Columbia University or somewhere that has one of these programs. Again, there's something like typically it's a 10 to 12 week over the summer. You are provided usually some type of support for room and board during that time, especially if you're going somewhere where you don't live, right? So I mentioned that I did research over a summer in Minnesota. I was living in Pennsylvania, right? So I had to go out and move there for the summer. Stipend for those 10 weeks, again, usually in the $5,000-ish range, and you're doing some type of hands-on research project in a faculty member's lab. So very similar is this NIH program. So NIH is the National Institutes of Health, right? So this is the more biomedical research. The NIH as an institution is outside of Washington, DC, right? So it's not a university, it's just a big research campus that has, the National Cancer Institute and the National Lung and Allergy and something Institute. Like they have all these different kind of areas within biomedical science. Same kind of time frame for the applications, same or similar kind of amount of money that you get, time frame in terms of how many weeks. This one is a little bit shorter maybe. And one other thing to mention, there are a couple of NIH programs that are not directly in the main campus, which is outside of DC. There's one in Framingham, Massachusetts. For folks who are local, that's the National Heart, Lung, and Blood Institute. So again, on their website, you can look and kind of search for different opportunities based on what your interests are. We have this Roger Williams Jobs Board, which I think was recently resurrected, maybe. So
Today.
Recently meaning today. So hopefully we will be adding a lot more opportunities to this coming up. But this could be jobs in terms of, short-term research things. This could be something, if you are getting ready to think about graduating and you're trying to find opportunities, those will be posted on here. This is the type of thing where as faculty, we have a lot of connections with people doing different types of research or different types of science, and they may email us and say, hey, I know you're a professor and you have all these people who are about to graduate. Do you know anyone who might be a good fit for XYZ? And we may reach out to particular people, but we will also post it on here. So this is a good way to find people who are looking for someone who may be in your position.
Yeah, I think the most recent additions to it were kind of in early May. I think the server was down over the summer, but like I said, we just got it started again. So as materials come through over our computer in terms of opportunities, faculty will routinely post it on these. I think actually I give an example of what one would look if you click. So if you're interested in that one, click. And then you can see it provides a lot more information about what the job or internship entails and how you go about applying.
Okay, and then one other thing I wanted to mention, this is a little bit different from some of the others. So there's these post-baccalaureate, meaning after you finish undergrad, post-baccalaureate research programs. You know, Professor Taylor mentioned how often it's ideal if you get started with research early, but you know, some of you sitting here may not be first or second years, and you might be thinking, oh man, is it too late? Is it too late for all these things? And it's not, it's never too late. Right. I, again, did one of these. I was a chemistry major as an undergrad and decided in like my senior year that I actually wanted to do biology research. And I had never taken a biology lab. So I'm not quite sure how I decided that, but apparently I did. And I went and did one of these post-baccalaureate research programs. So usually these are, you know, 1 to 2 year programs where you would go and essentially just your job would be working in someone's lab and doing research there. So it's really great to have that kind of full-time experience, right? And you get a lot more research done. You learn a lot more kind of about the day-to-day. It can be great if, like you're like me, you're trying to do something different, switch fields, or maybe you just didn't get into research as early. You know, maybe your strength is in the lab more so than on exams, and you're trying to show, hey, maybe my GPA isn't what I really wanted it to be, but if I can go here and maybe get a publication, you know, that can be a really great thing to put on your resume. So let's see. So a lot of the times for these post-bac programs, the expectation is that you are going to be applying to grad school or med school or some other type of professional school while you're there. And there are several of them. The kind of most centralized one is again at NIH, the National Institutes of Health, which is outside of DC. So you apply for that like the same way you would apply for like a school, right? You send an application to NIH, but then it's a little bit different because you would also be looking at individual labs and maybe emailing those PIs and saying, I'm interested in working in your lab. My application is already in the system, take a look. So that's one program that's out there. And then there are a lot of these programs called prep programs, which are kind of all around the US. I put the website there in case you want to look at them and kind of search through them. A lot of these are for underrepresented students, but not all of them. And prep just essentially meaning you're going to go there, you're going to do some research. You might have some other like classes or workshops that you're involved in, and you're preparing for kind of the next step.
All right, so that's what we had to present for you guys in terms of why, how, and what kind of opportunities exist for research here and beyond our campus walls. I hope you found it informative. Certainly, I'd like to open up if you have any questions, we can entertain those. All right, then I'm going to ask a question. So at the start, I said there are 3 things to keep in mind. What was number 1? You reinforced it over time. Start thinking about research opportunities early, right? You don't have to be a junior or senior, but oftentimes the good opportunities, the great opportunities come your first and second year. You get multiple years of experience. So start early. What was number 2? Absolutely. Be aware of deadlines, whether they are in a specific semester and in some cases a specific year. You get 1 crack at it. Okay. And what was the last one? Other than research experiences the full spectrum of human emotion. The other one was, I know this was a lot of information coming at you. Fear not, there are resources that are going to become available to you to see this again, whether it's in video format, or if you reach out to either one of us, or just go to the website, we can provide you PDF copies of all of this, plus the document that outlines the faculty profiles. Yep, sure.
So I'm a transfer student and I took a semester off. Would I still be eligible to sign up for program?
That is a great question. I don't know this with certainty, but what I suspect is your year class is going to be defined by the number of credits that you've earned. So if the university designates you as a sophomore, I believe you would be eligible for that junior and senior award.
Okay, so this semester would be my second semester as a sophomore.
Okay. Well, that's a great question. I think I'm guessing.
Okay.
So don't don't write this in stone. My guess is that the deadline is not, so whether you are a semester ahead, semester behind, the deadline for the Hollings is fixed. Regardless if you're gonna, like, where you are, you know, whether you're a semester or a semester late. So that deadline is fixed. My understanding is if you are locked in as if you identify as a sophomore, you are eligible. However, what would have to be considered is if it's a 2-year fellowship, so that means you're asking for a year and a half to graduate this semester.
Right.
That's a great question. I haven't seen that come up. We need to explore that.
A lot of times, if you look at the program's websites, they'll be usually quite specific about the eligibility criteria. But if they're not, a lot of times they'll give like a contact person that you could email to ask that type of question.
Thank you.
Okay.
Right, a coffee break. Right,
And do you want to stretch legs or just get started? Just do the
What's the timeline? Are we getting started?
5 minutes late.
Oh, no. Okay. No coffee break.
That was because it took me a little while to convert it.
But you want people to stay for the student?
No. So we're going to convert over to student presentations. I do want to talk to the presenters for just 1 second, so you can stretch your legs and then quickly come back. So that's 2 as well. Actually, I want to talk to all the presenters for a second. So I think most of you have seen it, but so this is going to be the end slide that operates all the presentations. So when you actually look under how it's different colored, your presentations are hyperlinked to that. So all I'm going to do is click on it, and then you'll off this is these are quite that's to advance forward, that's to advance back. So go back. So slide forward. So you should be all set. I will start the presentation for you. I'm not going to do any lengthy introduction. I'm just going to say Veronica's now going to be talking about scientific distribution, and then I'm just going to end it. And then you can talk about this. What I'm sorry. No. That's what I was going to say. So what I was going to ask is just coming off the heels of this presentation, if you wouldn't mind, if it's your first when you're talking, introducing yourself, just very briefly explain how you got involved in research and how you've been supported. Clearly, it says like who you work with, how did you get recruited to the lab, were you supported by grants, were you supported by cert fellowship, just so they can tie it into some of the things. Okay. Okay, cool. It's still recording now. Yeah. That's why it was a little bit of a delay. We had to put it over to the All right, so we're going to begin with our first student presentation. It's going to be Veronica talking about cyanophase distribution in the Atlantic Ocean.
Hi everyone, I'm Veronica Millett. I'm a senior marine biology major here at Idfr1. So first I just wanted to tell you how I got involved in research. So I'm actually a part of the Marine Science Club, and Dr. Marston had reached out to the club and said there's an opening in her lab and if anyone's interested to reach out. And so my way is a little unprofessional, but then I reached out to Dr. Marcy Marston and I was accepted and able to work in her lab. I've been working with her through last fall and spring semester, and then I got the opportunity to work with her through this summer, and I was funded by our NSF S-Score grants. And so I would like to start by saying the ocean is full of viruses. They can affect things from phytoplankton to large whales. And so they influence the marine food web dynamics. So viruses are able to infect the phytoplankton, which then they're able to lyse the cells, and that kills them. And so then that affects the whole trophic level from grazers and up to carnivores. They're also able to affect the abundance and diversity of host populations through their host range. So a virus is able to infect different types of bacteria. They can have a broad host range, which means they can infect many different types, or they can have a narrow host range where they have a very specific type of bacteria that they can infect. So for example, a virus can infect only the light green phytoplankton, or they can infect the yellow and the light green. They also, their viral communities are vary. They depend on the location and the host, and so their genetic composition can change based on the location in which they are, and then the host that they're able to infect there. And so the host that I isolated my viruses on was Staphylococcus. This is a small unicellular cyanobacteria, and it is very abundant in coastal seawaters. They are photosynthetic primary producers, and they can be red and green. And so then the virus that I isolated, they are cyanophage, meaning they are viruses that are able to infect cyanobacteria. They don't affect humans, and they have double-stranded DNA. They're also very abundant. Two families of viruses that I looked at were Myoviridae and Potoviridae. These are the myovirus and potoviruses, and they have very large DNAs. So we received our samples through, we work with University of Delaware, the Vail Lab, and the Hugh Sharp cruise went out on a 10-day research cruise off the coast of Delaware, about 140 nautical miles out past the Gulf Stream, and then they came back up towards Delaware Bay. On this cruise, they had 25 different sampling sites in which they took 4 different depths of water samples. And so you can see this is the course that they took, and all their sampling sites are highlighted. And so from there, we were able to form our research question. So our first one was, do the types of viruses differ depending on the depth and or sampling location in which they're from? And then from there, we thought, are the viruses that are found at different depths and locations, do they vary in their sets of genes that are in their genomes? And then our third question was, how does the movement of ocean currents influence the gene exchange among these viral populations? We're interested to compare our viruses isolated from off the coast of Delaware up to Narragansett Bay, Rhode Island viruses. So these are our methods, what we did to isolate our viruses. So first we started with our 92 unfiltered water samples. We got a box of them, and we isolated them on our red Staphylococcus bacteria cells, and we used Woods Hole 7803. We then formed enrichment plates using our samples and bacteria cells. So each plate is a depth, the 4 depths across one site. And so the red wells means that bacteria is present, they're happy, they're living, they're growing, and no virus was able to infect them. But when you see a clear well, like right here, you know that virus is present, it was able to infect the bacteria cell, and so lysis occurred, killing the bacteria. So then we wanted to isolate an individual viral isolate. And so we did that through plaque purification, because in your enrichment wells, we were using unfiltered seawater, so you could have many viruses in there, and you just want one to be able to sequence their DNA. And so on the plaque purification plates, they're pink from our cells, and when there's a clear dot on them after about 2 weeks of growing, we know that there's one individual viral isolate at that point. We take a toothpick, we pick it, and then we can sequence them using PCR techniques. And so for marker genes for the myovirus, we use the DNA polymerase gene, the G43 gene. And for the potoviruses, we use the ribonucleotide reductase gene. Those were our marker genes. And so from there, we were then able to create phylogenetic trees and do phylogenetic analysis based on these specific genes, and then sequence their full genomes and do genomic analysis. And so here are some of our first results. So this table shows the Cyanococcus infecting cyanophage across the locations and depths. And so across the 24 sampling sites, you can see all of the red boxes are where we were able to isolate individual viruses, and these are all myoviruses. The 2 green boxes are where we found photovirus. And so the sites 5 through 13, which are all blacked out, those were the sampling sites past the Gulf Stream, and we actually found no virus there that was able to infect our Cyanococcus cells. So here is one of the phylogenetic trees that I made. This is for the DNA polymerase gene for the myoviruses. This is a nucleotide-based phylogenetic tree. There are 120 viral isolates on this tree across all 13 sites in which we had detected myovirus. These fall into 23 different clades. And so a clade is isolates, and they're based on the amount of similarity they have. So these are highly similar, and they fall into a vertical line, like right here. So for 17 of these clades, the DNA polymerase gene was about 95 to 100% identical to viral isolates up here in Narragansett Bay. So that was very interesting to us to be able to see that we have similarity from off the coast of Delaware up to near us in Rhode Island. And so generally across our cyanophage populations, we saw that there were differences from site to site, but then we also saw there were some differences within one site. So now we were able to do some genome sequencing and make host range assays. And so our isolates were isolated on Cyanococcus Wilkesville 7803. We then took 5 other Cyanococcus cell lines, which you can see they're colorful, and these are controls on both right sides. And again, when you see the color, that means that they're growing, they're happy, there's no virus. But then when there's clear wells like here, you can see that virus is present and was able to infect and kill the bacteria. And so what this host range assay does is it tells us what bacteria strain our viral isolate can infect. And so some have very broad host range, like it can infect 4 out of the 6, like Cas1, or it can have narrow ones, which isn't seen, but they can infect only 1. And so we saw over the isolates that we sequenced their genomes that there wasn't much overlap in patterns. There were very different patterns from each isolate. And so for an example, the first one, Cas1, cannot infect this bottom cell line, but Cas2 could. And so now we wanted to compare our genomes. This goes back to our third question. And so we have here a viral isolate Cas2, which went off of the cruise, and then Rhode Island myovirus 143. And so this is their whole genomes. There's about 190,000 nucleotides. They are very large viruses. And so here it shows that they are very similar, about 94.4% similarity on the nucleotide level, but there are some genetic differences. So you can see here where the Gould spaces are, there are allelic differences among those genes. In blank spaces, genes were different, so either missing or gained between each other. And then or dark green, of course, nucleotide similarity at those spots. And so we were really interested to see how the genomes compared from down off the coast of Delaware up to Rhode Island. And so now this goes into some of our further studies. We want to look at how the ocean currents, including the Gulf Stream, are able to influence and affect the viral populations, and how gene exchange through these viral populations occurs based on how they're moving in the ocean, down from where the cruise went off right here up to where we are in Rhode Island. And we will also continue this fall to isolate and characterize additional isolates from our water samples and sequence more. We'll get more full genome sequences from some of our isolates and we want one from each clade to have a wider range of genomes. So that is it. I can open up the floor to any questions if there are any. Thank you.
So what do you think accounts for the lack of viruses in the Gulf Stream, but then you get coastal, anywhere coastal, you're finding that one. So what is it about the Gulf Stream that's?
So I think past the Gulf Stream, the abundance of Cyanococcus bacteria cells is not as high as it is near coastal waters. And so the viruses that we're finding out there, it's why maybe we didn't see any that infected our Cyanococcus cells, because the bacteria is not as abundant, so the viruses that would be able to infect it out there are as abundant. So maybe if we were to try something like Phlococcus, cells, which are more abundant past the Gulf Stream, we would find viruses that are able to infect that type of bacteria.
And then we have a poster afterwards.
Yes, and so if you would like to come see me at my poster, feel free to. I'll be out there.
I guess, so just to follow up that same question. In the coastal areas, you did have that one site that has nothing. Amongst all the, is that an error?
I think that one site, the sample in the box opened and we didn't have anything from that one. It was kind of the way it was transported for that one example.
So is there an optimum concentration or population size of Senecoccus that the viruses do best at, or does it have to reach a certain level, the population of Senecoccus, before the viral load starts to get hit?
I'm
just curious,
I'm not sure, but I think there has to be a high abundance of them in order for there to be viruses that are affected.
Just go ahead.
Okay.
Just work on the technical things. Great, thank you very much.
Thank you.
Okay, so our next speaker is going to be Cass. She's going to talk about some CRISPR technology and using it with rotifers.
Thank you. Hi everyone, my name is Cass and I'm a marine biology, chemistry, as well as aquaculture and aquarium science major here at Roger Williams. I first got involved in undergraduate research here because I basically just wanted to take a deeper dive into my major and also to get some real world experience as well. I started research by asking around the different faculty, and Dr. Ryan is the one that gave me an opportunity here to work with Dr. Bertner. And I'm supported through grants through my advisor, Dr. B, as well as I've gotten my own grants. I've gotten a Tri-Beta grant, which is an external grant from Roger Williams University, as well as the senior thesis grant and the Provost fund to assist in my research. So my research involves genetically modifying rotifers using CRISPR-Cas12a. And first, the question is, why rotifers? Rotifers are a great model organism. This is a picture or a video I took of our lab culture. They're really tiny. They're able to live in just a 12-well plate that we keep at 28 degrees Celsius. We feed them algae from our very own wet lab, and they're kept at a salinity of 22 parts per thousand. They're also great model organisms for genetic research due to their asexual life cycle, in which the female is able to produce an egg that develops into an embryo without fertilization. And most importantly, as far as genetic research, their genome was just recently sequenced in 2019. Rotifers are also a great source of food for aquaculturing larval fish. Larval fish will eat a lot of rotifers, and they're really easy to culture for these reasons. However, the nutritional value of rotifers is really lacking due to lacking taurine, which is a sulfonic acid. Larval fish need this to develop properly. So whenever larval fish are fed rotifers, that feed often needs to be supplemented with another source of taurine. A great way to go around this issue would be to genetically modify rotifers to implement the taurine biosynthesis pathway. However, this would include adding in 3 different enzymes into the rotifer genome, and that is quite a lofty goal as no one's done that before. So instead of this, we are going to utilize a different protein, which is the green fluorescent protein, and try to implement that protein into the rotifer genome, which would actually make them glow green. So this would just prove that someday we would be able to modify the rotifer genome. So in order to insert something into rotifers, we first need to create a break within the DNA. This we'll use CRISPR for, which I'm sure you've heard of before. It's often been compared to genetic scissors, which is a really great analogy because it's a nuclease that is able to cut DNA. So CRISPR is a ribonucleoprotein consisting of the Cas CRISPR nuclease as well as the guide RNA. the Cas CRISPR nuclease and the guide RNA come together and they're able to form a functional nuclease, which is a protein that is capable of cutting the DNA. There's a guide RNA that is involved in this complex, and that guide RNA basically tells the genetic scissors where to go within the DNA to create a double-strand break. And that is how we're able to choose exactly where within the DNA we can create this break. So where we are going to create this break to insert the green fluorescent protein is within the IGFR1 locus. The IGFR1 locus is the insulin-like growth factor receptor 1 locus, and this is a gene that has an aging phenotype. It's been shown in other model organisms such as mice, fruit flies, and roundworms that knocking out this locus has been shown to extend lifespan. So we're hoping for a similar result when we knock it out in rotifers. We've designed 3 guide RNAs to be able to guide the genetic scissors to the IGFR1 locus and create a double-strand break. These are at position 456, 543, and 561 within the IGFR1 gene. To test these guide RNAs, we tested them in vitro using a Cas12a digestion of a 518 base pair PCR product of the B. plicatellus IGFR1 gene. And as you can see on this gel, we have a control that has no gRNA, and then we tested the 3 gRNAs that we designed. The control has no cutting, as you can see by the 518 base pair PCR product there. And gRNA 456 showed the most complete digestion of the PCR product. So therefore, we will be using gRNA 456 as our guide for the CRISPR-Cas12a complex. Now, when you create a double-strand break within DNA, 2 things can happen. The first is the default correction pathway and is more common. This is when the DNA just comes back together and it results in a lot of insertion or deletion mutations and is therefore error-prone. The second is homology-directed repair. This is less frequent and it involves a donor template to come in and create a precise correction within the DNA to make it whole again. We will be supplying the rotifer cells with a donor template, so we are hoping for this outcome on the right. However, it is anticipated that since this is infrequent, it will happen with less frequency. So this is the repair template that we have designed to place into the DNA. On the left and the right, it contains 2 regions of homology with the insulin-like growth factor receptor 1 gene, which is that gene that we're planning on breaking. And it also contains 2 promoters as well as the enhanced green fluorescent protein. And that is that protein that we are trying to insert into the rotifer gene in order to cause the rotifers to fluoresce green. So how I'm going to insert all this information into the rotifer is through microinjection. First, I have to first collect the egg from the rotifer, which I do through a series of vortexing and centrifuging. Once I have the egg, I grab it with the holding pipette, which is on the left, and on the right is the injection needle. In the injection needle will contain lipofectamine, the guide RNA, CRISPR-Cas12a, as well as phylogenextran, which is a fluorescent compound, which will aid in a visual marker to tell me whether or not I have properly injected the egg or not. So this is the microinjection setup that I have downstairs. On the left here is the holding pipette that I control with this joystick. And this is how I cause suction with the holding pipette. And on the right is the injection needle, the joystick for the injection needle, and the femtojet, which allows me to actually inject fluid through the needle. Here is an egg that I have injected only with phylogenextran, which is just that fluorescent indicator. It doesn't edit the rotifer at all. However, here's very compelling pictures that I'm able to successfully inject a rotifer egg, because this picture is 2 eggs, one that I have injected with phylogenextran and one that is just a control egg, so I did nothing to it. And it is both a fluorescent light as well as a regular light. So here we can see very clearly that the injected egg is fluorescing and the non-injected egg is not fluorescing. However, when I turn the normal light off and I'm left with the fluorescent light, only the injected egg is fluorescing. I've done blank microinjections, which is basically when I just inject the egg with nothing at all, just to see if the rotifer can survive the egg chorion being broken. And I've done several injections with Phylogenetic. So I'm anticipating our percent viability to fall somewhere between 33% and 16%. So once we are able to inject with the CRISPR, lipofectamine, guide RNA, and the Phylogenetic, just as an indicator, there are two anticipated outcomes that we might be expecting. The first is no edit within the germline. This still involves the egg being injected, and the parental generation will have a mosaic effect of the adult rotifer. This means that some cells will be affected and in turn will be fluorescent green and others won't be. However, in this case, there's no edit within the germline cell. This would then cause the next generation to be wild type, as that edit did not make it into the genes to be passed on. The second outcome would be an edit within the germline cells. Once we inject the egg with this outcome, the parental generation would still have the mosaic effect, where some cells are fluorescing and some aren't. However, the germline cell would be edited, which would then cause the next generation to fluoresce completely green, as all of their cells would be affected, and would also result in a long-lived rotifer due to the deletion of the IGFR1 gene. So those are my expected outcomes, and I'd like to acknowledge Dr. V and the entire Molecular Longevity Lab for their help and support, Dr. Andrew Ryan for helping come up with this idea, as well as his knowledge towards rotifers and getting me involved in research, Dr. Amy Herbert for her guidance in choosing the correct microinjection needle, and the wet lab for their saltwater and algae and for always supplying me with those things. Any questions?
Just curious, just from a technical standpoint, what kind of learning curve was it to inject the eggs?
An entire semester. It was hard. I had to teach myself how to do microinjection, and the rotifer egg is comparable to the size of a human egg. It's about 100 micrometers. So it was very tough, and it took going through 2 different types of injection needles to get the right ones.
Now, is that viability between 33 and 60%? Is that, I guess, typical? Is that a
Yeah. Yes.
What would you say was like a difficult part in your research?
Definitely. Well, I've been doing research since winter of sophomore year, and I'm a senior now. So probably developing tolerance to obstacles was hard. A lot goes wrong in research, and it was hard to keep showing up every day with like a positive attitude at times, keep trying things, especially during learning how to microinject. But yeah. Any other questions?
The last oral presentation for today is going to be from Sandy. She's going to be talking about developing PCR assays to detect fish parasites.
Okay, so hi everyone. My name is Sandy. I'm also a senior here at Roger. I'm a bio major, and I've been working with the Aquatic Diagnostic Lab since my freshman year here. I got involved because I knew someone in the lab, but I also really was adamant about wanting to do research right off the bat. So I made sure to get involved and email right away. And luckily they had a position open for me. So I started working with them right away. And I was given this project the summer before my junior year, and it's going to be my senior thesis. So I've been working on it for a while now. So I've been developing a qPCR assay for these 3 fish parasites. And I know this title is a little overwhelming, so hopefully I can break it down for you a little bit better. So just giving like a 30,000-foot glance at this, just a bit of background and overview. These three parasites that I mentioned in the slide before, Crypticarian Irritants, Uranema Marinum, and Neobenadinia, are very common within aquarium systems, and aquarists usually have lots of issues with them. They can cause a lot of mortality in tanks, and they're very quick to infect. And unfortunately, it's a very late diagnosis for them. Usually you can only diagnose until the parasites are already seen on the fish, and it's a very expensive treatment. I have a picture on this slide of the giant ocean tank from the New England Aquarium. We've been working very closely with them on this project, and you can imagine if one of these parasites had an outbreak in this tank, how difficult it would be to treat it, considering the fact that it's 225,000 gallons of water. So what we're trying to do here is provide a resource for early detection of these parasites by testing the water instead of the actual samples from the fish themselves. This would also give us the opportunity to constantly monitor the tanks for the levels of the parasites, and would ultimately give a quicker, cheaper, and more effective treatment for the aquariums themselves. So let's get into the three parasites that I'll be looking at. So the first one is Crypticarian Irritants, which you'll hear me refer to as crypto. This one is one of the most common parasites in the aquaculture world. You can even have them in your home aquariums. So it causes white spot disease, which is also known as marine ick, which on this poor little clownfish over here are these little white spots. Each of them are individual parasites. So on that fish is the trophont stage, which is the feeding stage of it, but we'll be looking at the theront stage, which is when it's free swimming in the water column. It has the capacity to infect multiple different types of species, and it's usually found on the gills and skin of infected fish. The next one is Uranema Marinum, which I'll refer to as Uro. And this one is a little bit different than Crypticarian in the sense that it's an opportunistic parasite. This means that it's free swimming in the water column throughout all the stages of its life cycle and only attaches to a fish when it's given the chance. It's thought to be present in the background of all tanks, so usually there's a background level always present. And this one infests and creates ulceration in the skin and gills of the fish. So on this fish, this is what's causing this ulceration here. And eventually this can cause mortality in fish due to its erratic swimming and rubbing against rocks, which can cause secondary infections and ultimately death in these fish. Our last parasite is Neobenadinia, which I'll refer to as Neo. This one's a skin fluke and it's the biggest of the three. So if you're looking at this fish, it's all these little things on here. This one can survive for up to 15 days without attaching to a host. But when it does attach to a host fish, usually it attaches to the fin and then can migrate up to the skin or around the facial area of the fish. And this one also causes erratic swimming and hemorrhage, and like all the other two parasites, eventually mortality if not treated. So you might be asking what made us choose these three parasites to study. So there's a couple of reasons why. First of all three of these fish have these parasites have very quick life cycles, which cause quick infection and major mortality within these fish, which is something that aquarists obviously don't want to have happen. Then they also affect a wide range of hosts. So this means that it can infect multiple species within a tank. So when we're looking back at the giant ocean tank, there's tons of different species of fish in that tank. And most of these parasites can affect a majority of those fish in the tank. But the most important one is that all three of these parasites have a stage in their life cycle where they're free swimming in the water column. So if we're looking back at Crypticarian, this is its life cycle. As you can see here, the therons is the free swimming stage. So theoretically, that means that if we take a water sample from these tanks, we'd be able to detect each of these three parasites once we extract the DNA from that sample. So how did we do that? First, we started with positive controls that were taken from skin and gill scrapes from infected fish that were either in our university wet lab, or participating aquariums also sent us some samples. We were able to extract the DNA from those using a blood and tissue kit from Piagen, but eventually we moved on to using water samples because that's what we want to be testing in the future. So we took 1 liter water samples from the giant ocean tank at the New England Aquarium, and we filter them through this vacuum apparatus in a 0.22 micron filter. And then we extract the DNA from that filter, and then we'll be able to run it through our qPCR. So again, another huge glance at qPCR. So if you've taken genetics, you've probably been familiar with PCR. So just a quick differentiate between them. PCR is able to tell you if you're positive for something, whereas qPCR will tell you that you're positive, but also how much of that DNA you have. So this is just a picture of a gel that I've run. This one's a plasmid, so it's got that up there. But you can see that each of these signals means that it's positive for the DNA that we're looking for. Whereas when we're looking at a qPCR, this is a standard curve from one of my singleplexes for Uranema. All of these X's here are our samples, and it tells us based on this standard curve exactly how much DNA are in those samples. So both of these can be used for diagnostic purposes, but we're using a qPCR to tell us exactly how much of each parasite we have in each sample. There's also a couple other things you're going to need for qPCR. So you're going to have to design primers, design probes, and then you'll be using Supermix and your template DNA. So this is just a graphic that I found to help better explain qPCR a little bit. So if in your sample you have DNA that's positive for one of these parasites, what'll happen is the probe that we designed will attach to the DNA. Once it denatures and splits apart, our primers will also attach, and then it'll give us an amplification here that it's positive. So basically what you're looking at is if the concentration of the DNA, of the positive DNA is higher, you'll have a higher amplification. It'll come up in an earlier cycle of the qPCR. So our assay is 40 cycles. So if you have a super high positive for Crypticarian, let's say, it might come up in cycle number like 5. But if it's a low positive, it could come up in cycle like 35. So when designing this assay, we did base some of our work off of previous work done by Taniguchi et al. They designed a singleplex cybergreen assay for Crypticarian. We based that and made our other singleplex assays off of that and adjusted where needed. We were able to create standard curves for each parasite with plasmids, and we designed our primers for Neobenadinia and Uranema, as well as our probes for all 3 parasites. All of our primers were tested for cross-reactivity, meaning making sure that they're not testing positive for other things using BLAST, and we also confirmed in a melt curve analysis. So this would be an example of a melt curve analysis that we don't like. We had this one for Neo originally when we designed our primers, and you don't want to have multiple peaks here. So we're looking for something like this, which is what we eventually got when we redesigned our primers, which shows that there's no cross-reactivity and that everything is working well together. So as I mentioned, I'm a thesis student, so this project is still ongoing, but I do have some preliminary results. So we've designed singleplex assays for all 3 parasites, meaning that we can test water samples and tell you how much of each individual parasite we have. We've also run preliminary duplexes. So this one's a duplex testing for both Crypticarian and Uranema here at the same time. And we've also run a preliminary triplex, which is this pretty picture here. And so that's basically saying that we can test for all 3 parasites at the same time, which is what we're going to be looking for in the future. So we're still optimizing different primer and probe concentrations to make sure that we have the best assay that we could possibly have, but we do have these preliminary results, which is really exciting. So now you might be asking, well, how does this refer to the aquariums in our samples? We did do a bunch of testing with the giant ocean tank at the Nogan Aquarium, and we did get some data from them. We ran a bunch of their 1-liter water samples through our qPCR, and we were able to get some positive results, which you'll see in these bars here. And we compared that to what the aquarium has for their own scale for their infections. So the red of their scale is their highest infection, and then the orange, and then the yellow. And you can see that there's this really large disparity here of where we should be seeing the positive samples. So we figured, okay, giant ocean tank is 225,000 gallons, which is about 850,000 liters, and we're only sampling 1 liter. So there's probably something there where we're not testing enough water. So when we're going into our future work, what we're planning on doing is we're using these Waterra filters now that can sample up to 1,000 liters of water, which will give us a bigger sample size to test off of. So we're working with them and testing out different filtering methods to make sure that everything can be more accurate. And we're beginning to monitor the levels of the 3 different parasites with 3 different aquariums, so we can start getting that going and getting more research on that. And I just wanted to leave this presentation because I know that it's a celebration of undergraduate research and you guys might want to get involved. I have really enjoyed my 3 years working on research here with the ADL. I've had so many opportunities and made so many friends along the way. I've had the opportunity to present my research multiple times. I even got the opportunity to travel to San Diego and present at an aquaculture conference there. And I would just really encourage all of you to kind of branch out and figure out where you can do your research and just get that experience, because it's been really fun. So I can open for questions now.
So you mentioned about the complications of sampling the GOT, the ocean tank. Just a small volume of water relative to how big it is. Is there any evidence that the parasites might differ in their depth distribution as well?
We haven't really looked into that yet, but I'm assuming that could be part of it, because when we took the samples, it's from the top of the ocean tank. We haven't done any sampling with them with the bigger filters yet. We've just been using their quarantine facilities, so that's definitely something that we can look into. We're trying to figure out exactly where we're going to be putting the pump and the filter setup, so that's definitely something to consider.
Kind of a broad question. A lot of these parasites, so the bacterial, the third one's a fluke.
One of those skin fluke, yeah.
They're small. They're at least the flukes are on the surface of the animal that look like. How something like erratic swimming, you know what the actual mechanism is, or what they're doing that leads to that kind of.
I know for at least Uranema, it does have some neurological effects on the fish, and I'm assuming it's probably the same for Neobenadinia. So that definitely helps with it, but part of it is also just rubbing against the sediment to try to get the parasites off as well.
So if you like detected like the parasites in the water, would you be able to treat the water, or would you still have to treat like individual fish? Like how would you apply it?
Yeah, so that's kind of the reason why we started all of this. As I was saying at the beginning, it's not very cost effective to treat the giant ocean tank after a large outbreak has occurred, because what you're doing is you're either dropping the salinity, or if you have a smaller tank, it's like freshwater dips to try to take the parasites off. So what we're planning on having is, if we can detect the parasite outbreak before it happens, it's a cheaper treatment because they don't have to drop the salinity as low, and it's less harmful to the fish that are in the tank that aren't affected by the parasites. Yeah.
So do you think you will ever have the ability to answer the question of some level of parasite infestation in the water is acceptable, but once you get to this concentration, you have to treat?
Yeah, that's basically what our goal is, especially for something like Uranema, when it's kind of present in the tank at all times. Just trying to figure out at what level does it become a problem is one of our main goals. Yeah.
Thank you very much. So that concludes our oral presentations from our students. Let's please thank the student speakers one more time. They did a What we have next is a poster session. We're actually close on time. So we have a poster session out in the M&S lobby. You'll see various examples of more student research going on. I encourage you to look at the posters. Obviously, look at them for their science, science that's being done, but then also pick the students' brains about how they got involved in research and what different mechanisms they were supported by. It's a great idea or a great sense of to learn about how you get involved. I don't know if there's still cookies out there. They might be gone. Don't know they're gone. Never mind. So enjoy the empty tray. But please stick around for the poster session.
Good job, Cass.
So good, I got you.
Yeah.